Today we discussed about the various steps of DNA replication. We already know DNA are the genetic material for all living organisms except retrovirus. There are mainly three steps in the process of DNA replication. First, unwinding of DNA double helix. Unwinding of DNA double helix. Then the second step, synthesis of new DNA strand. Synthesis of new DNA strand. And the third, synthesis of primer. The first step, unwinding of DNA double helix. Second step, synthesis of primer. And third step, synthesis of new DNA strand. Now we draw a rough picture of DNA replication fork. DNA replication fork. It looks like a fork, so it is known as DNA replication fork. There are two leading strands. There are two leading strands in case of this fork. This is another leading strand. There are two types of strands, leading strand and lagging strand. Now the template, that is the leading strand. We draw the two leading template strands. Then we discussed about three different steps of DNA replication. First, unwinding of DNA double helix. Second, synthesis of primer. And third, the synthesis of new DNA strand. First step, unwinding of DNA double helix. The two strands of replicating DNA. The two strands of replicating DNA molecule separate by the action of enzyme helicase. The separation helped by the presence of an enzyme known as helicase. Then another enzyme, topoisomerase, keep it open. There are two enzymes. First, the enzyme helicase and second enzyme topoisomerase in case of DNA unwinding. DNA unwinding. Two strands of replicating DNA molecule separate by the action of enzyme helicase. Then another enzyme, topoisomerase, keep it open. Another enzyme, topoisomerase. Now complete the first step of DNA replication, that is unwinding of DNA double helix. Second step, synthesis of the primer. Second step, synthesis of primer. Primer is a short RNA molecule. Primer is a short RNA molecule of about 5 to 10 nitrogen bases. It is formed, that is, the primer is formed in the presence of enzyme primase. Primer is a short RNA molecule of about 5 to 10 nitrogen bases. It is formed in the presence of another enzyme, primase. Now we discussed about 3 types of enzymes. The primer provides a third and OH group for attachment of the new DNA strand. OH group for attachment of the new DNA strand. Now we draw the picture. Leading strand and the lagging strand pictures. This part is known as parental strand. This is the parental strand. Parental DNA strand. This is the leading strand. This is the template, that is the leading strand. And this newly formed one is known as lagging strand. This is known as lagging strand. These bits are known as Okazaki fragments. These bits are known as Okazaki fragments. Then we discuss about the third step. That is, this is the primer. Primer is now known as the Okazaki fragment. The synthesis of new DNA strand, that is the third step. The third step, synthesis of new DNA strand. That is the third step of replication. The DNA synthesis always take place from fifth to third direction. The DNA synthesis always take place along fifth to third direction. Fifth to third direction. Fifth to third direction. So the new DNA strand get synthesized in the direction opposite to the leading strand. This is the leading strand and this is the lagging strand. So the new DNA strand get synthesized in the direction opposite to the leading strand. The newly formed strand is known as lagging strand. By the help of enzyme DNA polymerase, new pieces of DNA are synthesized, now known as Okazaki fragments. Fifth to third direction. The new DNA strand gets synthesized in the direction opposite to the leading strand. The leading strand acts as a template. Here, one of the enzyme helps for this process, that is known as enzyme DNA polymerase. Enzyme DNA polymerase. The new pieces of DNA. The new pieces of DNA are known as Okazaki fragments. By the help of enzyme ligase and energy from ATP, Okazaki fragments joined together to form DNA strand. New pieces of DNA are synthesized is known as Okazaki fragments. Here is another enzyme help for this process, that is enzyme ligase. By the help of enzyme ligase, and energy is also needed. So this energy get from ATP molecule, adenosine triphosphate molecule. Okazaki fragments joined together by the presence of enzyme ligase and utilizing energy. The Okazaki fragments join together to form new strand of DNA. Now the replication of DNA is completed. If you like this session, please like, share and subscribe.